Well, Mendel knew a lot. He didn't know everything there was to know about genes. He had no idea about DNA or chromosomes. It's actually pretty remarkable that he worked out so much of inheritance so accurately. Today I want to talk about a couple of special cases that Mendel didn't cover. First, gene linkage. The law of independent assortment says that each trait is going to get assorted or mixed up independently from every other gene. Mendel was lucky that the traits he studied in pea plants worked out that way because he didn't know about crossing over in chromosomes. It turns out that crossing over is pretty good at scrambling up genes when they are far apart on the chromosome, but when those genes are right next to each other, we say they are linked. The chances that crossing over is going to separate them are very slim. For that reason, some genes tend to be inherited along with others and rarely separate. The other special case is polyploidy. By now you should be familiar with the terms diploid and haploid. Diploid cells have a double set of chromosomes, one from mom and one from dad. If meiosis goes wrong and an organism ends up with whole extra sets of chromosomes, that's fatal in animals. In plants, however, it can be a good thing. Having an extra set of chromosomes seems to give plants an edge. They might be bigger and healthier than other plants. A lot of crops we eat are polyploids, having as many as 6 extra sets of chromosomes. Gene linkage and polyploidy are both easier to study in organisms that aren't people. In fact, polyploidy is fatal in humans. The genetics of humans are harder to work out because humans don't have a lot of offspring at once. But as we learn more about DNA, we know more about ourselves and our inheritance. We'll talk a lot about DNA in the next unit. Stay awesome!